My name is Elizabeth Hénaff. I'm a professor at the NYU Tandon School of Engineering. So that's the branch of NYU that is here in downtown Brooklyn. And so my background is in computer science and biology. I studied computer science as an undergrad and then ended up getting a PhD in computational biology. And what I'd like to talk to you about today is some of my science research, but also some mostly my artistic practice. And so, I worked for a while in, my training is as a scientist. And as a scientist, we're really taught to remove the human or the individual out of our practice. That happens in a bunch of different ways. But for example, when we write a scientific article about our results, we always write in the third person. So like the experiment was performed according to XYZ conditions, not, I did this experiment. And so I think that there's perfectly good reasons to do that, to try to remove the human from the practice of science. One of them being is that we want to be able to rely on whatever we find out as reproducible results. So we want for the results of these experiments to actually be independent of the hands that are performing them. But as time went on, and I was working in biomedical research, I really started getting kind of bugged by the idea that really the practice of science is really indissociable from the people who are doing it. So, the experiments that we decide to do, the topics that we decide are interesting, the instruments that we build, those are all, made by people who carry in their own preconceived ideas, perspectives, hopes, biases. And so even though we try to really dissociate those two practices, they're very much intertwined. And so I realized that, that my practice as a scientist, that methodology wasn't quite appropriate or to start trying to tease out this relationship between science and culture. And so as kind of a different way of knowing, I decided, where I just kind of, started working on art projects that were kind of addressing the similar types of topics that I've been addressing as a research scientist, but just using these different methodologies. And so what I wanted to talk about today are some of these projects that have emerged from different research, or some of these art projects that have emerged from these different research projects that I've been working on. And so in general, I'm interested in how organisms interact with their environment, and that kind of thread has really been a driving factor throughout my career as a research scientist. And so I'd like for us to take a moment to maybe close your eyes and conjure up a mental image in your mind of the environment. Like, what do you see in your mind's eye when you think of the term the environment? Does anybody have anything that they would like to share about the environments that they've envisioned? You can unmute or you can drop it in the chat. Maybe Monica, if you want to monitor the chat for me. When I think of the environment, I think maybe of something like this. So this is a still from an anime called Princess Mononoke that has been produced by Studio Ghibli. And so when we think of the environment, maybe we think of, lush green forests, jungle landscapes with, plants and animals. Maybe a human being, maybe a human makes their way into our concept of the environment. Maybe not. That's another question for a much larger philosophical discussion of why oftentimes we don't perceive humans as being part of the environment.
So just in the chat right now, I think atoms, how do you say that in English?
Oh, okay, cool. So
I think nature, and I think of a forest.
Mm-hmm.
I think of ever.
Did anybody have a human in their environment?
I built environment in balance with nature. No.
No humans. Yeah, it's surprising. I've asked this question many times to many different audiences, and it's very rare that we get humans in our concept of the environment. So, you know, when we imagine the environment, we imagine oftentimes things that we can see, and usually living organisms in our environment, plants, animals, forests. But what it turns out is, you know, a ubiquitous and very abundant and diverse aspect of our living environment we actually cannot see. So, you know, in just, you know, 1 teaspoon of soil of this jungle landscape, we would have billions of microorganisms. And what I've been focusing on the last few years is studying in particular this living component of our environment, this invisible living component of our environment, which are microorganisms. And what's pretty exciting is that within the last decade or so, we've had advances in DNA sequencing technology that allow us to study these microorganisms in completely novel ways. So we've known about microbes for a long time, but most of what we know about microbes is by growing them in a petri dish. You know, so you like take a sample and you swab it out into a petri dish and you put it in an incubator at a particular temperature and you wait to see what grows. But there's a lot of microorganisms that do not like growing in a petri dish. We call them recalcitrant microorganisms, which I think is just really funny. It makes me think that they're like grumpy or something. and so there's, you know, there's this strong kind of methodological bias with respect to what we can know about microbes if we can only know things about microbes that are willing to grow in petri dishes. But DNA sequencing is a technology that allows us to extract molecular and genetic information from a sample without having to go through a step of culturing. And so this is a much more comprehensive and unbiased way to understand, you know, what kind of species are there and what they're doing. And it turns out that knowing things about microbes is really important if we want to study other kinds of microorganisms. So it turns out that there's a lot of like physical characteristics or, you know, phenotypes of multicellular macroscopic organisms like plants, animals, ourselves, that are dependent on the interactions that we have with microorganisms. So for example, it's been shown that you can alter the flowering time of certain plants by altering the microbial composition of the soil in which they're growing. So this was kind of a big deal in the plant biology world because flowering time was thought to be a like well understood, canonical, genetically controlled pathway. So different plants have different strategies for reproduction. So some of them flower, you know, before the cold weather, after the cold weather, at different seasons. And so that was thought to be a like strictly genetically controlled pathway. And so the realization that this, you know, phenotype or physical characteristic was due not only to the genotype or genes encoded by the plant, but also the genotype of the microorganisms with which it's living in symbiosis and cohabitation, was really a big deal in the plant biology world. And so this is the case for plants. This is one of several examples, but it's also the case for mammals, including humans. So we're pretty familiar with the idea of the gut microbiome being a really important factor in human health, and that disturbances in the gut microbiome are correlated with different states of disease. But it turns out that these interactions with microorganisms can have a lot more kind of subtle consequences, including that of mental health and well-being. So it turns out that the synthesis of serotonin, which is a neurotransmitter that acts as like an information messenger in the brain. That the synthesis of that particular molecule is controlled by microorganisms in the gut. And so this is another example about how the phenotype or physical characteristics of a macroscopic organism are determined not only by their own genotype or the genes that we encode, but also the genotype of the microorganisms with which we live in symbiosis. And so when we consider the notion of identity as being defined by genetic identity, the fact that our identity is linked not only to our own genes, but also the genes of the microorganisms that we interact with, this kind of begs like a redefinition of the notion of identity to kind of broaden both the organism, but also its symbionts. And so there's a term that has been coined for that, which is the term of holobiont. So the term holobiont encompasses the notion of both host and symbiont to redefine the notion of an individual. So we can talk about the human holobiont, which then includes, the human and all of our associated microbes. We can talk about the coral holobiont also. So, the coral body itself and all of its associated microbes. And so similarly, as we can talk about, use the term genomics to describe the study of the genome of an organism, we can also talk about hologenomics as the study of a genome of a holobiont. So including both microorganisms and the individual itself. And so the projects that I've been working on in my lab are centered around this idea of holobionts. So trying to think about the microbiome as a continuum between organism and environment. So at NYU, I run the Laboratory for Living Interfaces. And so there we study the interaction of organisms and their environment through both scientific and design inquiries. So the idea is that if, the human body, if, humans and human health and well-being are affected by our interaction with microbes, so the human microbiome, and then the human microbiome can interact with the environmental microbiome. And as, residents of urban spaces, the environmental microbiome is very much sculpted by the design decisions that we've made for the built environment. Then ultimately, microorganisms are kind of a continuum between human and environment. And so we have a whole portfolio of projects where we try to understand specifically what is the relationship between the built environment and the built environment's microbiome, with the idea that, if the human microbiome is affected by the built environment microbiome, and the built environment microbiome is ultimately designed through our design interventions, we should ultimately be able to design like probiotic buildings. What exactly we should be optimizing for is a whole bigger question. And specifically, I think it's hard to be prescriptive to decide what kind of microorganisms we should be designing for example, our indoor spaces. But there's a whole field of research that's addressing that. And so what I'm interested in particular is this interaction between the built environment and its microbiome. So for example, we're working on a project studying the Gowanus Canal microbiome. So this is the microbiome of the sediment that has accumulated at the bottom of this Superfund site, which I'll talk more about in just a second. We've also been looking at how climate change related events affect the urban microbiome. So specifically looking at the impact of street level flooding on the microbiome of sidewalk surfaces. We've also been looking at how green wall infrastructure, so specifically indoor green walls, can be kind of tuned to inoculate indoor spaces with specific microbial species. So what I'd like to do today is talk about how these projects have inspired artworks. specifically. So I'm glad to talk about the science behind this. If anybody is interested at some point, today or another day. But today, really, I would like to talk about the art interventions that I've done, kind of inspired by this. So for those of y'all who live in Brooklyn, which is maybe most of us, we're probably all familiar with the Gowanus Canal. And so the Gowanus Canal is one of two Superfund sites that we have here in New York City, the other one being Newtown Creek, which is at the border between Brooklyn and Queens. And so I was particularly interested in the Gowanus Canal because when we're trying to think about the interaction between microbiome and environment, when we're studying this at a city scale, it's hard to get interpretable information because these are such big open systems. So say, for example, we did a project where we looked at the microbiome of the New York City subway. And so we took a bunch of samples in all of the different subway stations in New York City. And so that was a good map making exercise, but it's hard to establish causal relationships when the system is so open. So, if we observe certain microbes in a subway station, could it be because of the human occupants? Could it be because of the fact that it's underground? Could it be because of the airflow? Could it be because of the pizza rat who's been dragging a slice of pizza around? It's just a really big open system, so it's hard to establish kind of cause and consequence relationships between aspects of the environment and aspects of the microbiome. But if we look for what we call extreme environments, so environments that have really strong selective pressures, we're able to tease out better these relationships between environment and microbiome. So when we think in extreme environment, we think maybe, lunar landscapes or the high altitude salt flats of the desert of the Atacama, or the North Pole or the South Pole. And so, we tend to think of these like exotic, faraway environments, but there's actually, we can actually find extreme environments in our backyard. And so that's what prompted this particular study in the Gowanus Canal, was looking for an extreme environment that was like actually easy to get to and easy to sample. And so what we did is we've been running this longitudinal study where we take samples of the sediment at the bottom of the canal. So the canal used to be a creek. It was dredged in 1853. So this, this environment was inhabited, is stolen land. It was inhabited by the Lenni Lenape people, and used to be a very productive estuary, like marine estuary. And then as part of the violence of colonization, these people were displaced as kind of the first act of violence, and then subsequent acts of violence include the dredging of this canal to serve as a means of transportation to and from the industries that were in operation around that site. And so it also served as like a de facto dumping site for all of the industrial waste that was being generated. And so today, the canal has accumulated about 10 to 15 feet of contaminated sediment underneath the water. And so we've been studying the microorganisms that inhabit this sediment. And so what we do is we, go out in our canoes. We work with the Gowanus Canal dredgers, who are a group who go boating in this extreme environment for fun, and who help us with the logistics of the boating and the sample taking. So we collect these samples of contaminated sludge, take it back to the lab, extract DNA, sequence it, and then ask the question, okay, what kind of microorganisms are living there? And what are they doing? And so it turns out that not only is there a kind of diverse and thriving microbial population in this environment that has been, decided to be as unenvironmental as one can imagine, but also these microorganisms have encoded or have evolved these really interesting bioremediation functions. So over the last 170 years now, they've had the time to be able to develop kind of molecular mechanisms to counter the toxic compounds that they've been challenged with. And so we found that the Gowanus Canal microbiome encodes a whole host of degradation functions that allow it to degrade the toxic compounds that it's been challenged with. So for example, it can degrade cresol, which is a petrochemical byproduct, arsenic, a heavy metal. It can fix arsenic, which is a heavy metal, degrade toluene, which is an industrial solvent. And so what's interesting is that, when we look at this list of bioremediation functions, we can correlate each one of those metabolic functions with historical data of the factories that have been in operation over the last 170 years. And so in a certain way, the present day 2021 microbiome of the Gowanus Canal maintains a historical record of the history of human intervention at that site. Arguably, maybe a better kept record than human kept records, because records are notoriously flawed, and history is written by the victors, and so this might even be a better kept record of the anthropogenic interventions in this particular location. And so in a certain sense, it's like a molecular echo, if you will, of the history of human intervention at that site. And so so this work is ongoing. Actually, we were supposed to be going sampling later this afternoon, but it's raining today, so we had to postpone that. So this work is ongoing, and what I think is really kind of a few concepts that I've distilled from this project is that the valence or qualities that we give an environment are very, very human-centric. So, the EPA, the dredging that's happening in the canal right now, the EPA has designated it as a Superfund site, and they have started remediation processes. So they're dredging out all the toxic sediment and going to cap it with concrete and put the water back and call it all good. And so from a human perspective, this environment is like the most unenvironmental environment that you can think of. We've just got to make it go away in whatever way we can possibly imagine. But from a microbial perspective, this environment is actually a perfectly amenable place to live and thrive. And if they were left to their own devices, these microorganisms would very slowly but surely decontaminate this particular environment. And so from a perspective of engineered landscapes, this intervention that's being done in the canal at the moment, so this dredging and capping is like a very intense mechanistic intervention in this environment. And if we were to imagine that intervention in maybe one of the environments that we imagined at the beginning of this talk of our lush green forests and junglescapes, that would be considered like a very violent intervention in an environment. And so I think what's interesting to think about is that if we try to kind of decenter the human with respect to our kind of like judgments that we make of different environments being good or bad, then we realize that actually we might act very differently. And so one of these projects that I've been working on has been trying to think about kind of interfaces of collaboration with these microorganisms. And so this is the first in a series of zines that I made that I've called Gene Zines. And so this particular issue is related to the Gowanus Canal. And so what's particular about this zine is that there's DNA that I've extracted from the sediment that is embedded in the paper of the zine itself. So these are just block printed kind of book-like objects. and with DNA that's embedded in part of the paper. And so the idea, I was inspired by kind of zine culture as being these like self-published small kind of packets of information that are aimed to a particular to distribute information within a particular subculture. So zines are often related to particular bands or different music genres, and so this particular zine is aimed for as a genomic note from the underground, serving the subculture of multi-species collaboration. And so the idea behind this interface and the kind of collaboration involved in it is that you can kind of tear out the part of the paper that contains the embedded DNA, and then seed that in an environment that's in need of remediation. And so this is based on the fact that microorganisms are able to absorb DNA from their environment. So we're born with the genes that we got, and we have to use them our whole lives. But microorganisms are able to just swap in new genetic information and just start using it. And so the idea is that the microbiome of the canal has had 170 years to adapt to the particular cocktail of toxicity that it's been challenged with. And so you can use this kind of molecular memory to serve a recently contaminated environment. And so you can, embed this piece of paper in a recently contaminated environment, and then the local microbiome would be able to use that that kind of generational knowledge from this other microbiome. So this was one project. I've also done a lot of projects involving kind of map making. So making maps of environments from a microbial perspective. And one of the challenges with making these maps on an urban scale is that we're operating at very different scales. So when we take a sample with just like a swab, which we're now all uncomfortably familiar with after having had a swab stuck up our nose like way too many times. But when we're taking a sample with a swab, we're taking a sample of a very limited area. And so if we're trying to study something as big as a city, there's a real kind of disjointedness between the scale of the instrument that we're using and the scale of the thing that we're studying. And so this is a project that I worked on with a bunch of collaborators a few years ago, where we decided to see if we could use beehives, so urban beehives, as like citizen scientists, kind of as samplers, because we realized that. So this was after a project that we did in the New York City subway, where we swabbed every single subway station in New York City over the span of the summer, and it was extremely labor intensive. So we had just these armies of undergrad students running around the subway with their swabs and like collecting all of these samples. And we were like, oh man, that was so labor intensive. Like, is there any way that we can do this in with like a higher, like a not higher resolution, but like a broader tiling, like a broader resolution? And we realized that bees are doing exactly what we're asking our undergrads to do, which is basically go out into the world, interact with a bunch of stuff, and then come back to the same place. And so we ran this pilot experiment that was led by a student who was an undergrad at Cooper Union at the time. Now she's a PhD student at the MIT Media Lab, Devorah Najjar, who fearlessly made a bunch of contacts with local beekeepers and piloted this experiment. And so we realized that if we collect the debris at the bottom of the hive, so all the pieces of just kind of plant material, dirt, dust that accumulate at the bottom of the hive, we're able to get this microbial fingerprint of an environment. And so we ran a bunch of pilot studies, and ultimately, so we performed this study in a bunch of different cities around the world. One of the cities where we had the highest density of hives was in Tokyo. And so what we were able to see is that the material collected from the hives gives us kind of like a quantified fingerprint of these different neighborhoods. So what we're looking at here on the right is a heat map. So this is a data visualization of the diversity and abundance of microbial species in these different locations. And so the way that we read this is that a yellow color means a high abundance, and a green color means a low abundance or absence. On the left here, on the y-axis, we have a whole list of microbial species. And then on the x-axis here, so each one of these columns corresponds to a different location. And so we can see, say here, we take these first few columns on the right, they all have very similar patterns, and those correspond to locations that are very close to each other. So the hives that are very like physically near to each other. And then if we look at this rest of these columns, this is another group of hives that are near each other, but that are farther away than the other set of hives. And so what this showed us really was that these bees are collecting like a microbial fingerprint of the environments that are within their foraging radius. And what was interesting is that, so what we first thought, we were like, okay, cool, we're really just going to get plant-related microbes because bees are only really physically interacting with the plants that where they're foraging. But it turns out that we recovered microbes from a bunch of different sources. So, you know, human-related microbes, but also, say, aquatic-related microbes, if there happened to be a body of water within the foraging radius. And so what we realized is that what we were observing here really was that these bees were kind of traversing these, what we call microbial clouds. So this is a term that was coined by Jack Gilbert from the University of Chicago to kind of explain the fact that, you know, that a microbiome is like a continuum. So I can talk about, you know, my skin microbiome and the microbiome of the table that I'm sitting at, and we can, you know, measure those two things, but actually like it's kind of a continuum between the two. It's you can imagine it like measuring temperature. Like I can measure the temperature, you know, inside of my house and then outside in the street, and so I get 2-point measurements. But actually, that like metric of temperature exists at like all points in between. And so what's happening really is that these bees, so we all have, you know, our like microbial cloud. So kind of this like, yeah, this like cloud of microbiome that corresponds to, you know, our particular bodies, or like my dog's microbial cloud, or the microbial cloud of a pond or a river. And so what we realized is that these bees were traversing, even if they weren't physically interacting with these different elements of the environment, they were traversing all of these different microbial clouds as they were foraging, and then recovering that material as they came back. And so we presented this work at the Venice Biennale, the Venice Architecture Biennale in 2016, in which we installed actually a beehive in the gallery space, which was a feat of engineering to like build out this, you know, have them be able to come in the window, and then we like build out this whole box for them. And we showed visualizations of the data that we had collected in the 5 different cities that we had studied at that time. And so this project was kind of like a map making exercise, and there's a lot of such map making endeavors that are being done by the scientific community at the moment, which I think is kind of the first step in trying to understand our relationship, or the relationship between the built environment and microbiomes. But something in particular that I've been really interested in is in addition to map making, or maybe beyond map making, is thinking about what would it look like to design for microorganisms. And so this is a project that was shown at the Storefront for Art and Architecture in 2018, which is a collaboration with Kevin Slavin, who was a professor at the MIT Media Lab at the time, and David Benjamin, an architect here in New York City who leads the studio called The Living. And so this was a commission by the Storefront for Art and Architecture, in which the curators asked us to imagine what would it look like on an architectural scale, so on a human scale, if we were to design for microorganisms. And so I don't know if y'all are familiar with this particular space, but the Storefront for Art and Architecture is a building that has very porous boundaries. And so each one of these panels that you can see is a door that can swivel open. I think I have a better picture here. Yeah, that swivels open, and that allows access to the space. And so what we did was try to think about, yeah, what does it look like on a human scale if you design with microbes in mind? And so it's kind of a mental gymnastics of like translation of scale in space, because microorganisms are very small. They exist, you measure them in microns, and human beings are relatively orders of magnitude larger. You measure them in centimeters or meters. And so part of our approach was thinking, not thinking about shapes in design, but also thinking about materials. So, what kind of material could we use that would have bioreceptive properties, so properties that would welcome or host microbial populations. And so for that reason, we settled on using wood as a material, being that given that trees are intrinsically a bioreceptive material before they became 4x4 posts at Home Depot. And then kind of the design intervention that we did was to fabricate these panels that kind of became part of the facade. And then with these panels were constructed with like wood tiles of like a cross section of a 4x4 post that had been cut and mounted, and then sandblasted. And so what the sandblasting does is that it kind of reveals the intrinsic three-dimensional structure in these wood tiles, because the softer part of the wood in the circles, in the kind of yearly circles, is degraded faster than the harder parts of the wood. And so in that way, we were kind of creating these microclimates, or these kind of micro three-dimensional environments in this bioreceptive material. And I think something that was interesting to me is that particular intervention felt very reminiscent of the practice of microscopy. So, if you have your sample and you're preparing it to look at it in a microscope, and it's really small and it's on a tiny flat little slide, and then you put it in the microscope, and then you look in, and then all of a sudden you realize that there's like a whole world in there that had been invisible before. And so through that instrumentation, you can see mountains and valleys and all of the structure that previously had been flattened and invisible due to the difference in scale. And so for me, that process of sandblasting was kind of doing a similar thing with these physical objects, and that it was revealing a three-dimensional structure that was intrinsic to the material, but that previously had been obfuscated. And so we also set up like a full-blown lab in the gallery space, where we were sequencing samples that we were gathering from this bioreceptive material as the exhibit went through. And so what we see here, I think still play, is a drawing robot that had been programmed to draw out letter by letter all of the nucleotides of the sequencing data that we were gathering. And so, when we're thinking about scale, we were kind of going through these like explosions and implosions of scale. And so, we were building something on an architectural scale, but taking samples of something that was microscopic, and then, boiling that down to just, a tiny little drop of water, and then getting data out of that. And then the data is actually explodes into a huge scale after that. I mean, from a particular sample, you can gather hundreds of gigabytes of data, and so what we were really interested in is kind of illustrating that the like large scale of the information that we were gathering. And so at the end of the exhibit, we just had sheets and sheets of these big mylar, or like these big mylar sheets that had been inscribed with this genetic data set that we were gathering. So this was a project where I was trying to think about kind of this like translation of scale in space, and about how when we think about interacting with microorganisms, we really need to do this kind of mental gymnastics of being able to think through these different orders of magnitude in space. And that's also the case in time. And so, microbes exist at a time scale that is completely different than our own as well. So like a microbial generation in optimal lab conditions lasts for about 20 minutes. So in the time that we'll have been speaking together, there will have been 3 generations of microbes that will have succeeded themselves just in this hour. And so this was a project where I was trying to think through this difference in time scale, and how can we think about different ways of measuring time, and of visualizing cycles that exist beyond our time scale. And so this is a tide clock that I made, and that was prototyped here in the Hudson River. And so the way this works is that it's actually a buoy that's anchored to the bottom of the river, and this is like a 2-meter tall PVC, or not PVC, acrylic tube that fills up with water at high tide, and then as the tide recedes, that level of water is maintained inside the buoy, and then when the tide gets completely low, it flushes out. And then next tide coming up does the same thing, fills up with water again, and then maintains that level of water until low tide. And so this was a project that was installed in Japan at Okayama for the Okayama Art Summit, that was curated by the artist Pierre Rig, whose work also oftentimes involves other kind of more than human species. And so this particular installation was accompanied by another edition of the Zine, in which I was looking at the microbiome of 2 adjacent bodies of water. So this body of water that we see here, that was a river, and then right behind where this photo was taken, there was a moat that circled around a like 12th century castle. And so that moat had been originally filled with water from the river, but then those 2 bodies of water had been separated for now centuries. And so I did another edition of the Zine, looking at the microbiome of these 2 bodies of water, and how you could see traces of evolution happening at different speeds in these 2 bodies of water. So in general, what I've really been thinking about these days is this idea of interface. Like when we're trying to build a relationship with an organism that we can't see, ultimately the relationship is always with the kinds of instruments and interfaces and interventions like that we create to interact with that invisible organism. And so for example, so the object very much dictates the nature of the relationship that we have. So on the left here, we see a swab, which we're now, as I said, all very uncomfortably familiar with. And so this is an instrument, an object that was designed to take a microbial sample, so to collect microbial cells for study. And this object kind of dictates a very specific kind of feeling or relationship. You know, it looks like a clinical object because it was designed to be used in a hospital, and it makes us think of contagion and disease and pathogenicity. But really what that object is doing is exactly the same thing as the image on the right. So on the right, you know, gathering a handful of soil is also taking a microbiological sample. But the image on the right has a completely different valence. Like, it makes us, you know, think of, you know, connection and groundedness and earthiness, and it has all of these kind of like positive feelings associated to it. And so what I've been thinking about really is trying to tease out what is the relationship to the object or to the instrument, and then what is the relationship to the organism that we're studying. And there's a lot of really interesting writing about this topic. So there's, you know, the whole field of study of kind of material culture is, you know, a field of study that addresses this particular idea of objects as being manifestations of ideas and relationships. Karen Barad is an author who I admire a lot, and who's a physicist and also a feminist theorist. And so she's thought a lot about the relationship that we have to instrumentation as a way of knowing. And so in particular, she writes, you know, and I'll quote her: "Apparatuses are not inscription devices, scientific instruments set in place before the action happens, or machines that mediate the dialectic of resistance and accommodation. They are neither neutral probes of the natural world, nor structures that deterministically impose some particular outcome." Then she goes on to say, importantly, apparatuses are themselves phenomena. And so I think this idea, I'm really interested in exploring further this idea of the ongoingness of our relationship with microorganisms, and how that relationship has been mediated through different apparatuses and phenomena over time. And I think in particular now, in the time of a global pandemic, when we're all very acutely aware of this invisible biological component of our environment, and that realization feels very new. It feels very out of the ordinary, and we've developed all sorts of interfaces that kind of manifest that relationship. So, you know, the masks that we wear, and the new rituals that we've developed of, you know, hand washing when getting home, maybe the anxiety that we feel in close proximity to another person in an indoor space. And so all of those, I think that kind of acknowledgement of the fact that there exists an invisible kind of biological metric or dimension to our environment feels very new. But I think what I've been interested in thinking about now is trying to think through the facts that we have never not interacted with microorganisms in our environment. And that relationship is one that we can trace all the way back to the origin of, you know, evolution of multicellularity. Even like multicellular organisms like ourselves, we evolved in a microbial and viral world. So there's never not been an interaction. And so that kind of idea of ongoingness is what I've been thinking about these days. And so I've been trying to think through, you know, what are examples of that ongoing relationship that we can see that exists, you know, before and beyond this kind of like Western biotechnological relationship that we're very much immersed in now. And so one example of this is this phenomenon of bioprecipitation. And so there's a hypothesis that rain dances work through this phenomenon of bioprecipitation. And so rain dances are native practices that have been practiced all over the world for as long as, you know, as human memory exists. And so there's this hypothesis that they work specifically as a kind of technology that enables interaction with a very specific kind of microorganism called Pseudomonas Syringae. So Pseudomonas Syringae is a microbe that produces a protein that's able to nucleate ice crystals at warmer temperatures than that nucleation usually happens. So ice is you know, the solid form of water, and ice crystals usually form by growing on a particular particulate matter. So say when we're talking about cloud formation, they will form around like a piece of dust or a piece of ash that is in the atmosphere. So these microbes produce a protein that is able to initiate the formation of those crystals at warmer temperatures. This is the protein that's used to make fake snow. So like on ski snow slopes, you have those like snow cannons that make fake snow so that people can ski, even if it's like early in the season or late in the season. They use this particular, like a synthesized version of this particular protein. And so there's this hypothesis that rain dances, which last for days, will kick up this particular microbe that lives on the surface of plant leaves. So they'll kick up that particular microbe, but then after, you know, a little while, floats up to the right part of the atmosphere where it's able to nucleate ice crystals, so like make clouds, which then lead to rain. And so I think, you know, when we're thinking about this, the ongoingness of our relationship with microbes, we need to think kind of beyond the like techno-scientific object of the like scientific instrument, but also think of interfaces like rituals, for example. And this idea of modern Western technology being the gatekeepers of like technology is totally wrong, and that there has been very ancient, accurate technologies that have been developed that enable these interactions. Another idea of this ongoingness with in our relationship is also with respect to pathogens. And so this is a map that was made in 1854 by Dr. John Snow, where he made a map, took a map of the city of London and colored in the houses that had a case of cholera. And he was able to, with this like data visualization, was able to identify that what these houses had in common was access to a particular well, and hypothesized that the well, that particular well, was the source of contagion. And then upon closing access to the well, was able to curtail that outbreak. Also, one of the ongoing relationships that we have with microbes is with respect to food and food fermentation. And so this is the oldest bioarchaeological record that I was able to find in the published literature that demonstrates the use of microorganisms for food fermentation. And so this was found in Sweden and was a gutter dug in the rock that was full of these fish bones, but fish bones that had been really squished, that had been deformed. So these are the fish bones on the right compared to fish bones of similar fish alive today. And so the hypothesis is that this deformation could have only happened in an acidic environment. And one of the hallmarks of fermentation is that the fermenting microbes create an acidic environment, which is what allows them to proliferate and prevents other spoilage microbes from growing in that same environment. And so This record was dated to 9,000 years before the current era, and is the first bioarchaeological record. So this was at a time where in the, say, Mediterranean basin, where domestication of plants such as grains was just starting. And so in a certain way, this was a different strategy for food storage and survival that involved domestication of microorganisms, just like domestication of plants and animals ensured the ability for food storage in other parts of the world. And what's interesting to me is that what the remnant here is the vessel. And so, this particular trench dug in the rock was a vessel that created a microenvironment that was amenable for food fermentation. And fermentation is a very ancient practice. And what's interesting to me is that oftentimes when it's described, that process of transformation or the power of transformation is attributed to the vessel itself. And so even before there was kind of a mechanistic understanding of the existence of microbes, there was a very clear kind of understanding of like cause and consequence. Like I put this material in this vessel and then this transformation happens. And so this was a book published in the early 1700s that I found at the British Public Library that was describing the process of transformation. And so specifically, it talks about how these different processes of fermentation are a function of the vessel that you put in it. And so as such, we've had a long ongoing relationship with in creating these objects that allow us to mediate this relationship with this invisible living world. So that's all I had to share today. I teach a whole class around all of these ideas at NYU called the Microenvironment Exploration Lab, where we'll go through this idea of like microbial interface, and then students will work on physical like design projects. And we always participate in the Biodesign Challenge, which happens at MoMA every year in June, which is happening online this year. And so if y'all are interested in these ideas, there's really great student projects from universities all around the world. And with that, I will thank all of my collaborators with whom none of this would be possible. And I thank you for your attention. Oh man, I went way over time. I'm so sorry. I thought this was going to be a short one.
This was super cool though. I'd rather have you talk than me talk.
I'm glad to take questions. I also want to be mindful of everybody's time. And so if you want to reach out by email, that works too. But
I'm just curious about your gene zine. Just to get a little bit of clarification. So you took the genetic material of some of these organisms that have been transformed by the human elements, like toxic chemicals, et cetera, and then changed by the nature of evolution, and then squeezed them onto a piece of paper, and like, so I could like throw it in a polluted lake, and then maybe it would do its magic,
Yeah, I mean, surprisingly, paper is a really good substrate for storing DNA. And we use that on a regular basis to like exchange DNA between labs. Like you just take a piece of water paper and, just pipette your DNA solution on it and you let it dry. And once it's dry, it's extremely stable. And then you can just rehydrate it and then it'll, detach from the paper. And it's great. You can just mail it. And it's actually, I'm glad this talk isn't being recorded, but it's actually a really good way to get by biosecurity measures because you can just mail DNA like on a postcard or like in a letter, as opposed to sending test tubes that then you have to like register with your appropriate government and whatnot.
Interesting. Thank you.
Sure. Any other thoughts, reactions, questions?
So one more thing that that those panels that you're focusing on sort of the sculptural aspect of them, but the browner areas, those were organisms working on the parts that were more exposed, the underlayers were exposed, and that's why they were brown.
Yeah, so there's a couple of things going on there. One of them, so the panels, there's some areas of the panels that were sandblasted. So
Yeah.
that was like a mechanical intervention in the material of the wood that then kind of revealed the concentric circles, or turned what was like a 2-dimensional concentric circle into a 3-dimensional structure, right, with, you know, the harder part of those circles, I guess, corresponding to growth in the winter months when there's less water, being more resistant to the sandblasting. And then those were left out for about 6 months and then sampled for microorganisms. And that's what we were sequencing.
Okay, so it wasn't like that was causing it to be brown necessarily, but they...
No, that was just like good old weathering.
Okay.
Outside.
Well, this was fascinating. It's amazing. I mean, I've started to see more and more of this bio influenced art or inspired art in you know, shows around the world, and I'm always kind of squinting at it for a long time, going, what am I looking at? And and so it's really great to have sort of an insider's view and, you know, into the whole idea of it. It's great. Thank you.
Thank you.
Yeah, do you usually get a chance to talk about that to people that, you know, see your art? Because this was, you know, fantastic to listen to you for the last hour. So, you know, when you...
Yeah, I mean, I think that's the challenge with installations, is that, you know, it's like a mad rush for opening day, and then there's like a bunch of people there, and then that's kind of it. And so I think it's nice to be able to have this kind of opportunity to kind of tease out the conceptual basis of the projects. Because you're right, as you were mentioning, Lily, I think a lot of these projects can be really uninterpretable, which to me feels like that was like an unsuccessful art project. Because I think an installation should speak for it, or should be able to have a voice of its own without having to read like pages and pages of explanations. And maybe that voice and that interpretation is saying something different to the viewer than what was originally intended, which I think is fine. But I think that is one of the challenges with this intersection of disciplines, is that in the sciences, we're used to having a lot of means for self-explanation, and I think a successful art piece is successful without a lot of explanation.
That's interesting.
Which I don't claim to have done successfully, by the way. I'm still working on that one.
Yeah, it's just how big do you want to make the plaque?
Yeah, exactly. How long do you want to spend, like, you know, pulling the like the insides of the D's and the P's on your like vinyl on the wall?
I just
Go ahead.
What's interesting is this all seems very logical. It doesn't seem like way out there science. It just seems like very commonsensical, you know, like it makes sense, of course. As you're saying it, I'm going, well, of course, you know, it's quite straightforward.
I don't know, the rain dances through me.
Really?
Yeah, because it's like, that's always something I thought was like a myth, you know.
I didn't think of it before, but then when you say it, it's like, well, of course, that's very simple.
Mm-hmm.
You know, you're making dust and it gets in the atmosphere.
Yeah.
Form and it rains. You know, it's like, okay. Or like, like all the buildings with biological, like plant walls and things like that, be having these effects on people. It's just like any kind of or like homeopathy or something. It's all kind of very basic in a way.
Yeah, I mean, I think what's interesting about thinking about traditional practices is that, they are, or they can be really reliable from like a cause to consequence perspective without necessarily the same understanding of the mechanism.
Right.
So,
if something works, you keep doing it.
Right. Mm-hmm.
But there was one question that I had when you said, like, just about the science of it. So if the DNA reproduces so fast or like changes so fast, when you said like in 20 minutes you have like 3 stages, right? For example. But then also, you can really study such a long history of something. How, that kind of threw me a little off.
Yeah, absolutely. I mean, so a short generational time doesn't necessarily mean that there has to be many changes in that short time, but there can be. So like microbial populations adapt to environments a lot quicker than other organisms with longer generation times. But if the environment stays the same, which in the case of the Gowanus Canal, it has been that contaminated for the last 100 years, then the populations there, once they've adapted to that environment, are going to stay pretty stable as long as the conditions aren't changing.
It's fast. So over time then, do you think, I mean, it will probably take a long, long time, but would like the Gowanus Canal be able to kind of, become something else that would, be less contaminated for a long time without our interventions that brutal that, that happen right now?
Yes, I mean, the question is the time scale, and also I think, and also there's kind of, there's a threshold in which, so also I guess a threshold of concentration. So, things like petroleum or petrochemical byproducts, tar, these are all naturally occurring substances that are found, in like large reservoirs in different places. And so there's, one thing is underground reservoirs and what their relationship is with microorganisms there. But, you can think of like, there's like open air tar pits that exist and that have existed for thousands, millions of years. And that despite the fact that that there is the possibility for interaction with local microbes, they're still there. So I don't think that we could imagine the Gowanus Canal microbiome making it all go away, for the reason that that there becomes a point where it's like no longer a challenge, and that actually there's kind of a cohabitation with that environment that happens, and then that just becomes a stable system. But that the microbiome of the Gowanus Canal could decrease the concentrations of toxic compounds to something lower than they are now, I would say yes. But make it disappear completely, I would say no.
Isn't there some question about whether dredging it is kind of dangerous in and of itself?
Yeah, I mean, there's several things that happen in response to that. One of them being that it's disrupting a lot of the material. Another question being, well, what do you do with the dredged material after that. I think at the moment it's going to be used as an additive for concrete. So basically being cast into concrete and used for other structural projects. And then another question is, once you cap everything with concrete, how stable is that? And that's kind of the standard approach for that particular kind of configuration, as determined by the EPA and the Army Corps of Engineers. And that's been implemented in other places, which is usually successful in the short to medium term, but then in the long term, there can be like, cracking of the concrete and then leaching of the substrate again. But I acknowledge that there's all sorts of different forces at play and different priorities and different timescales also. And so, when thinking about the hazard of human exposure, and that being a very real hazard, there's families who have been living in the Gowanus area for generations and who have been exposed to those hazards. And so I think it's easy to maybe be romantic about the idea of, if we left to their own devices, microbes would clean everything up. But there's also a really, a very real reality of the fact that this is an environment that's embedded in a very dense urban area, and that there are folks who have been exposed to these hazardous materials. So, and then it's also, a huge feat of civil engineering to think through about what you would need to do to dredge this environment and to cap it and to reduce that hazard of exposure. So I think it's a complex problem, and I think that at the time scale that we're operating on now, even though it seems frustrating because like as soon as the canal was declared a Superfund site, in a short amount of time, property values went up like 100%, and a lot of people were displaced due to that, and properties being bought up by developers with the idea that then they would sell waterfront condos on the canal. So there's a lot of things that are existing in tension there at the moment. And, I think unfortunately the people who have been historically exposed to those contaminants are now being displaced by gentrification. And so those families are not benefiting from the cleanup efforts that are at play at the moment, which is the case for most Superfund sites in general. So in general, I think there's a statistic like within a 1 mile radius of a Superfund site, you tend to have historically underserved communities in a bunch of other different ways. So there's a lot of intersections with both social justice concerns and also environmental justice concerns.
Thank you. I'm going to sign off, but thank you. Very great.
Yeah, very interesting. Wonderful. Thank you so much. Beautiful work and amazing.
It's really amazing. There were actually a few more questions, but I honestly have to also sign off. But this was amazing. It's really good, and I wanna meet you at the canal at some point, because I'm there very often too, and I'm working with the dredgers on some children program.
Cool.
And
maybe we can keep in touch, and next time when we finally manage to arrange a sampling expedition, maybe we can meet up then.
Right. Yes, that would be great. But thank you so much, Elizabeth. That was amazing. So if anybody wants to ask one last question, please go ahead. But I
just want to say thank you so much.
Yes.
It was super interesting.
Yeah, and you explained something. It's really like Rebecca said. It just makes it all seem so logical and so understandable. Like it's I'm sure I'm sure it's pretty advanced what you were talking about. Yeah, but you made it pretty understandable to us. So that was fantastic. Yeah.
Thank you. I appreciate that feedback. I think that has been honed by many generations of students.